Okay, you should have just completed Module 4, which covered genetics and mitosis, meiosis. And what we're moving into now is the last module, the preparation for your last exam of the semester. And this one talks about DNA and protein synthesis. In this first topic, topic 41, we'll be introducing nucleic acids. Now, really, you've already touched on nucleic acids as they are one of the 4 groups of biological compounds. And so we just want to kind of brush up on this, learn a little bit more about DNA, and then we'll move into DNA replication and also protein synthesis. So first of all, note that we are in Chapter 8. It's not quite in the same order that the book goes along, so we're kind of doing things a little bit out of order. So remember that we're now in Chapter 8, and we're going to talk a little bit about DNA specifically. DNA stands for deoxyribonucleic acid. Okay, so that's where the letters DNA come from. As I said, it is one of the nucleic acids, the nucleic acids are one of the 4 classes of biological compounds, and DNA is one of the most important ones of the nucleic acids. The monomer, the smallest unit of a nucleic acid that maintains its identity, is what we call the nucleotide, and we'll talk about that more specifically in a few minutes. The third question here, who discovered the structure of DNA? Watson and Crick were 2 scientists that came up with the model, the structural model of this double helix, twisted ladder type structure in 1953. And as you read the chapter, you'll find, of course, there were other people working on it at the same time. There was some controversy about who really first came up with the structure and then who recognized it for what it was. But basically, Watson and Crick are given accolades for this discovery. Here's a picture of them from your textbook, and a little bit hard to see the double helix here, but this is a pretty big model, very specific detail model of DNA. Here they are again. Watson was only 23 years old. 23 years old. As I've probably said before, sometimes, it's a miracle that I even make it to work every day. But this guy was on top of things and, came up with this at age 23. Crick was 35 years old, a former physicist. Watson, a former ornithologist, which, in case you don't know, that means he used to study birds. Before we go on, I do want to mention the Human Genome Project, and this was an international science project that scientists were working all over the world on this, and it began in 1990, and the goal was to map all the genes of a human in 15 years, and it actually got through a couple of years early. So what they were trying to do is identify the order of the nucleotides in all the chromosomes of a human. They didn't just choose one human, they actually used 4 that were chosen randomly and anonymously, 2 males and 2 females, and sort of put those together because, of course, everybody's genome is different in some ways, but were sort of generalized genomes of the human. And even today, it's considered one of the world's largest collaborative biological projects. Pretty amazing how this was all done and that it actually took place as quickly as it did. The major findings from the Human Genome Project were that humans have a lot fewer genes than were expected. It's always been thought that the more complex an organism was, that the more genes they would have, the more nucleotides to code for all these complexities, and that was turned out not to be the case. At the same time, they also found that there were more repeated sections of DNA than were expected, and this kind of relates to the idea that there are a lot of switches and stop genes, start genes, things like that, that were repetitive and not actually ones that code for proteins. This is a picture of Watson in 2007, and because he was one of the two men that came up with the structure back in 1953, they used his DNA to be the first person to get his own personal breakdown, his own personal genome map, which is sort of cool. And I think he is still living today as of October 2017, but he was 79 at this time in spring of 2007. Oh, we got to have a silly joke. This was 1991, so right after the Human Genome Project was completed. We finished the genome map, but now we can't figure out how to fold it. Ugh. Okay. Okay, so why is the structure of DNA so significant? The reason, what you have to know, first of all, is the structure of DNA consists of these nucleotides. And how many different nucleotides are there? 4. Only 4. The genomes, the chromosomes, the DNA of every living organism, whether it be a blade of grass or your own DNA, is still made up of these same 4 nucleotides. And so, in that sense, it explains how life can show unity at the molecular level, where all our DNA is all the same, just made up different arrangements of these 4 nucleotides, yet so much diversity at the level of whole organisms. Again, it's the order of those 4 nucleotides that make all the difference in you and a blade of grass. So what does the DNA code mean? Really, the DNA, again, it contains instructions to produce all proteins. Proteins are basically very important they're considered the stuff of life. They are what make up the majority of our body parts. They are very important in structure for things like collagen. They are carriers. In other words, example of hemoglobin carries oxygen. Also enzymes, all those metabolic processes that we have understood. Oh, let's just say you learned. I don't know how much you understood. I hope you understood it all. But anyway, all those metabolic processes require enzymes, and those enzymes are proteins produced by those genes made up of DNA. Another important concept here is that all this information in your chromosomes really is just like a storage place for this information. In other words, the genetic information is stored in your chromosomes, in the nuclei of your cells. Unless it is converted into proteins, it is only a storage place and means nothing. This is sort of a silly analogy, but it's very true. I'm not a great cook, but when I go through a magazine or I read the newspaper and I see a recipe that looks delicious, I tear it out. Oh, I got to do this. And so I have a huge drawer full of recipes for very delicious dishes. Of course, if they stay in the drawer and I never cook them, I never prepare those dishes, they're pretty much meaningless. And this is how you can sort of understand the role of DNA. DNA is the recipe, but you've got to have the proteins. You've got to have the delicious dishes, or they're basically meaningless. And so what we're going to learn in the last part of this chapter is how the DNA will code, how we get from the DNA to these proteins. And of course, a very good name for this process, protein synthesis. The other thing you should understand, and we've kind of gone over this before, is that the code is the same in all the cells of one organism. Of course, unless there's a mosaic situation, which we talked about in the last unit. But basically, if you took a cell from your cheek, from your eye, from your big toe, and looked at the chromosomes in the nuclei of those cells, they would be identical. However, certain genes, certain portions of those chromosomes are, let's say, turned on, activated, so that only certain ones actually produce the protein in those cells. My example here is a little bit silly, but it's hopefully makes the point. In the iris of your eye, you have cells and the chromosomes in there. There are genes, for example, for eye color. There are also genes in there that will code for keratin, okay? But fortunately, in your eye, the gene that it would be activated might be the one for melanin, the brown pigment, so that your eyes would be brown. In your big toe, you have the genes for that melanin. You also have the genes for the keratin. The one that is activated in your big toe might be for the ones that produce the protein keratin to produce a toenail. So again, it's scientists are learning a little bit more, and we'll get to this at the end of this unit, about why some genes are turned on in some cells and some in another. And that again is one of the really miraculous ways that the human genome and all genomes of all organisms do work. Okay, I've used some pictures from some other textbooks as well as from yours, but this is sort of a simplified one. Here's your basic double helix. Now we look at it a little bit more closely and we can see that it's sort of like a twisted ladder. Now we've straightened out that ladder over here, and some of the things that you can see are the side portions here, what we could call the uprights of the ladder, or whatever you want to call it. These are made up of sugars and phosphates. You have a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate. And the sugars, in this case, are actually deoxyribose. You may recall that OSE often signifies a sugar. In this case, yes, it does. And then these areas right here that make up the, what would we call those? The rungs of the ladder, the steps of the spiral staircase. These are what we call nitrogenous bases, and we'll look at those a little more closely in a minute, but they do have nitrogen in them, and there are four. They're abbreviated here with a T, an A, a G, and a C, and you'll notice that the T can only connect with the A, the G only with the C, and so forth. Okay, these nitrogenous bases, that is really how the nucleotides differ, and we'll talk about that more in just a second. Also, I want you to notice that the way that these nitrogenous bases are connected in the middle are by hydrogen bonds. Hydrogen bonds connect them. This is a good place to show you this as well, even though I don't know, I never ask you to know that adenine, for example, is a double ring structure, and thymine, abbreviated by the T, is a single ring. But I do want you to notice that that is one of the reasons that they combine as they do. There are always three rings across. 1, 2, 3. 1, 2, 3. And this makes the uprights an equidistance. Instead of the ladder going in, out, it's just straight up here and here, and it's always the same distance across. So that is something to note there. Here's a picture from your textbook showing the same thing. Again, you have the double helix. As it gets more magnified, we can see a little more in detail about the sugars, the phosphates, and the nitrogenous bases. These are pictures of the four different nucleotides, and what they differ in is the nitrogenous bases. The rest of this, the phosphate and the sugars, are identical on all of them. And you don't certainly have to know the difference in the structures of these, but again, you should know that we have adenine, guanine, thymine, and cytosine. You don't have to know that thymine and cytosine have a single ring, adenine and guanine have a double ring. What you do need to know is that adenine can only connect to thymine, guanine only with cytosine. And again, this is just one of them in, total detail for you to see. There's nothing that you specifically have to know. Actually, I see I have an error here because I have that this is DNA. Well, I might as well mention it to you right now. The difference in RNA, which I will get to on the next couple of pages, is that the sugar in RNA is not deoxyribose, it is ribose. So I kind of made a mistake by putting this particular picture in here because this is RNA, but we'll get to that in just a minute. Again, a total lie here. I say each one. It is in DNA. It is deoxyribose, but this is just the wrong picture because this is RNA. There you go. Okay, but anyway, the nitrogenous bases adenine, guanine, cytosine, and thymine. Those portions are the same in both RNA and, well, in DNA. Those are them. We'll talk a little bit about the difference in the thymine and the uracil in just a minute. Okay, now's a chance for you to be an artiste. And I hate to tell you, this isn't a very good picture, but it took me about, 4 pieces of paper to get it right. So I know this can be more challenging than I meant it to be. It's harder than I thought it would be. But anyway, again, simplified. You don't have any detail on the nitrogenous bases here, but just realize that one nucleotide is made up of one phosphate, a sugar, and a nitrogenous base. Those are the 3 things that make up a nucleotide. So this area that's circled, one nucleotide. On the other side, I have labeled the different parts. These are the nitrogenous bases here, the T, the G, the A, the C, the T. These little black balls here are supposed to represent the phosphates. And this little structure, I will tell you when I drew it, I finally figured out to sort of make it, to figure out how many points I had on it. It looks like a little house, okay? It looks like a little house, and that made it a little bit easier for me to draw, if that helps you at all. So again, in DNA, the sugar is going to be deoxyribose. And what connects these nitrogenous bases are hydrogen bonds. They're in the middle. Okay, if you need to stop this to finish your drawing, go ahead and stop it, but I will keep it going. Okay, this is just a little review of things that we have really already gone over in the previous module. Again, chromatin is often described as the DNA when it is all strung out, okay, when it has not been supercoiled into thickened bodies. So that's what we usually use the word chromatin for. A chromosome is, in biology, often considered the supercoiled form of it. So by the time you have mitosis beginning, they're nice and thickened, and you can actually see the 2 sister chromatids on there with proper magnification. Still one chromosome here, another chromosome here, even though because the DNA is already replicated during the S phase of interphase, we have the 2 sister chromatids double the DNA material itself. We also already have learned that human cells have 46 chromosomes, which is 23 pairs, 1 set from the mother and 1 set from the father, except of course in the eggs or sperm, and those are the result of meiosis, where you have an egg or a sperm with 23 chromosomes, just 1 set. And now we're going to add to this a gene. And basically a gene is considered a segment of DNA that codes for a specific protein. It's not actually quite that simple, but we're going to stick with that story. Our terminology, we're going to stick with the idea that you have 1 gene, 1 protein. Again, as technology has improved, as the body of scientific knowledge has increased, that is not always exactly true, but for the level that we're trying to understand it in this class, it'll work. Okay, now before we move on, I want to tell you a little bit about RNA. And again, it's not that I'm just trying to mess with your head, but it just makes more sense to me. We just talked about DNA. Let's talk about RNA here. In your book, there's this one little section here in the 9th chapter They skip on to the 9th chapter to tell you this. So just be aware of that. Okay, RNA is ribonucleic acid. Again, what's the difference? As I mentioned on that other page, the sugar. In DNA, it's deoxyribose. In RNA, the sugar is ribose. What is the difference structurally? Guess what? Deoxyribose has one less oxygen. Aha! So again, the name does make sense. Okay, and what we're going to learn when we learn about protein synthesis a little bit later is RNA is the middleman in the conversion from DNA to protein. So in protein synthesis, what we're going to find out in a nutshell is DNA, then you have RNA, and then you have your protein. And that is a pathway that is pretty much set in stone for most living organisms. Okay, so we're going to go through the 3 types of RNA. DNA is just DNA. There are not 3 types of DNA. There's just DNA. It's made up of 4 different nucleotides, but it's still basically all the same. RNA, on the other hand, there are 3 different types. We're only involved with the first 2. Okay, the first one is called messenger RNA. It's often abbreviated with a little m and then RNA. And I'm telling you its role. It's kind of like if you have ever read, say, a play, and at the beginning you have the list of characters and a brief explanation of who they are and what they do. That's what we got here. Until you hear the story, it's maybe a little bit meaningless, but it's important to get all this down in one place so that you can go back and refer to it if you need to. Okay, so what messenger RNA is, it literally acts as a messenger. It takes the code from the DNA and the nucleus, and it will go out of the nucleus through a nuclear pore and find a ribosome out in the cytoplasm. And again, we'll talk much more about that in a little while. It is generally a long strip. It is a half a ladder. Okay, so again, these sides over here, this portion right here along this part, that's going to be made up of sugars and phosphates. The little parts sticking up here are going to be the various nitrogenous bases. And what we call these three in a row, these triplets of 3 bases, is a codon. And again, we'll review all of this as we go through the notes. Okay, the second type of RNA is called transfer RNA. What does it transfer? It transfers amino acids, which, as I'm sure you remember, amino acids are the monomers of proteins. Okay? And so they're going to carry amino acids to the messenger RNA at the ribosome. Again, we'll get to the nuts and bolts of this. I started out here drawing the transfer RNA in kind of a tricky way, so it kind of even looks like a T. It was too hard for me. In the later drawings, I have it more like this, like a figure 8, and then this part right here will have the nitrogenous bases sticking off of it, and here I have a too thick a pen here, but in there I put AA, so it's a little easier to draw than this thing. But anyway, these 3 nitrogenous bases right here have another name, the anticodon. And again, we'll go through all of this as a review in just a minute. The third type is called ribosomal RNA, that, along with the proteins, makes up the ribosomes. Because it doesn't play a huge role in the story that we're learning in this section, don't worry about it, but know that it does exist. So those are the 3 types of RNA: messenger, transfer, and ribosomal. You need to be most aware of the messenger RNA and the transfer RNA. Okay, so now I have a little table here so that we can really compare these. Okay? Okay, and we've already been through all of this. The nucleotide is made up of a DNA molecule. The nucleotides are made up of sugars, phosphates, and nitrogenous bases. The sugar in DNA, deoxyribose. The nitrogenous bases, adenine, guanine, cytosine, and thymine. Okay, before we go, well, let's keep going. Let's keep going down. The structure, it's a double helix. There's only one type. And where do you find DNA? In the nucleus only. The only time that it is not really in the nucleus is during a mitosis division or meiosis division where the nuclear membrane dissolves. Normally, that DNA remains in the nucleus and is protected by that nuclear membrane. Okay, what are the differences in the DNA and the RNA? First of all, and most obviously, the sugar. The sugar in RNA is ribose, thus RNA. Deoxyribose, DNA. Okay, you got your phosphate, you got your nitrogenous bases. This you need to make note of. Adenine's the same, guanine's the same, cytosine's the same, but in RNA, the thymine is replaced by uracil. Slightly different structure. RNA has no thymine, instead it has uracil. Okay, it is always a single strand, meaning like in messenger RNA, you had that nice long strip like that for messenger RNA. For transfer RNA, you have what I'm simplifying as that, only 3 on there. And the messenger RNA, it moves, it's a messenger. So it goes from the nucleus out the nuclear pore to the cytoplasm, whereas the transfer RNA is only found in the cytoplasm. Okay, I have a little insert right here that again, you've probably already caught on to because I mentioned it before, but the A with the T, the C with the G. And the saying that I learned years ago, pretty stupid, is at the golf course. So A with the T, G with the C. I usually think of it as AT is, well, it's a word for heaven's sake. So the A goes with the T. The C with the G, they kind of look alike. You add a little thing to that C and it looks like a G. Okay, so that's just another way, whatever it takes for you to remember that. We will find that if we're attaching RNA to DNA, don't worry about this too much yet, but remember RNA, where you might think thymine would go, no, if it's RNA, it's uracil. And we'll go over this later again, but the T with the U, I think of those letters are next to each other in the alphabet, for one thing. And also you might think of UT, okay? So those are the ones that replace each other, depending on if it's DNA or RNA. Okay, here's a picture from your textbook showing some of the differences. And again, not extremely helpful, I wouldn't think, but it does say with the DNA, well, it doesn't even really show the sugars at all, but it does have deoxyribonucleic acid, ribonucleic acid. The first difference would be in the sugars themselves. And then the other is the thymine versus the uracil. And it also shows, this would be messenger RNA because it's a long strip, but that the DNA is the double helix, whereas the RNA is really just half of that. Okay, so let's finish up with this with a random multiple choice question. Which statement is correct about the differences between DNA and RNA? Okay, basically these are like a series of true-false questions is what it really means. Okay, A, the sugar in DNA is ribose. No. The sugar in DNA is deoxyribose. B, DNA always remains in the nucleus during protein synthesis. That is true. DNA never leaves. C, RNA is a double helix. No, it is not. DNA is a double helix. RNA is always just one half of that. D, DNA contains cytosine, guanine, uracil, and adenine. No, because DNA would have thymine, not uracil. E, DNA is the bridge between the genetic code and a protein. No, it is RNA that is the bridge. Okay, so this finishes up the introduction. Now we'll move on and talk about some of the processes. So this was a really important chapter to understand. Make sure you read your textbook on this material, and then we'll start next on DNA replication.